Assalamualaikum warahmatullahi wabarakatuh. My name is Nur Fazlini binti Saifuddin, and in this video, I will present the final year project on title Antibacterial Activities and Phytochemical Analysis of Ethanol Extract, Ethyl Acetate Fraction, and Butanol Fraction from Dalle Indom Fruit Shell. Begin with introduction of this study. Most of plants, including fruits, play a vital role in human life and commonly used for drug extraction, as mentioned by Talukdar et al. On 2022. In this study also, the fruit that was used, which is Xylem indum, locally known as Keranji, is particularly valued for its phytochemical, health benefits, and also antibacterial activity, as reported by Osman et al. On 2018. Moving to the problem statement, there are several issues or problems that contribute to this study, such as food waste and drought-resistant bacteria. Xylem in the fruit shell are often underutilized and discarded, contributing to increasing food waste. According to Silva et al. On 2018, fruit shells may contain bioactive compound, antibacterial, anti-inflammatory, and antioxidant properties. There are two main objectives of this study, which are to determine the antibacterial activities of Dalle indom fruit shell extract, ethanolic, ethyl acetate fractions, and butanol fractions against E. coli and S. aureus bacteria. Other than that, it's also to identify the phytochemical components that present in fruit shell ethanolic extract, ethyl acetate, and butanol fractions of Dalle indom. The significance of study are to develop a sustainable system to manage fruit waste and efficient use of fruit shells. Other than that, the study aimed to raise consumer awareness about the nutritional value of the Indian fruit shells, encouraging the use of entire fruits and minimize the waste. Other than that, the global search for new antibacterial drugs drives research into the potential use of the Indian fruit shell as an antibacterial agent. For the literature review, Xylem indum, or locally known as Keranji, can be found in tropical region as mentioned by Osman et al. On 2018, and this tree can grow up to 40 meters in height. For plant phytochemical profiles, according to Sheh and Patel on 2020, phytochemicals are naturally occurring chemical substances in plants, and there are several types of phytochemicals such as carotenoids, polyphenols, isoprenols, alkaloids, flavonoids, tannins, saponins, and terpenins. For bioactive compound in Xylem indum species, many different kinds of phytochemical and antibacterial properties can be found in various parts of Xylem species, such as Xylem guarniae, and these have shown phytochemical components that may be the source of the fruit good antibacterial and wound healing abilities, as reported by Santi and Sergetuel on 2016. For methodology in this study, start with sample collection, sample processing, sample extraction, sample fractionation, and continue with phytochemical screening and antibacterial tests, and lastly, statistical analysis. For sample collections, 1 kg of Dalle indom fruits were purchased online from Shopee platform. Continuing with sample processing, the fruit shell were dried in the oven at 47 degrees Celsius for 24 hours. After that, the process was continued by sample extraction, where the dry fruit shell were ground into a fine powder. After that, it was undergo maceration process by using 95% ethanol. And then, the solvent was filtered by using a Whitman No. 1 filter paper, and the solvent was evaporated at 40 to 50 degrees Celsius, and as the result, the dried indom ethanolic root extract was stored in a universal bottle. For sample fractionation, Xylem indum ethanolic crude extract was mixed with ethyl acetate with 1:3 ratio using a separating funnel. After that, the upper layer of ethyl acetate was collected, continued by the remaining aqueous solution, undergo fractionation using butanol, and then the solvent were dried in a fume hood at room temperature, and as the result, dried Xylem indum ethyl acetate fraction and Xylem indum butanol fraction were stored in a universal container. For phytochemical screening, this test was performed on D. indum extract DIECE and fractions DIEAF and DIBUF. This test was observing distinct color and physical changes using standard procedure at concentration of 10 mg per ml. This test was performed for phenol, flavonoids, saponin, tannins, and alkaloids. For antibacterial tests, treating this paper, gentamicin was used as positive control, while 1% DMSO as negative control, dilute in the ethanolic crude extract, dilute in the ethyl acetate fractions, and dilute in the butanol fraction were used at the concentration of 100 mg per ml. The disc was positioned onto the plate and compressed using forceps. After that, all the disc was positioning, the lid was replaced and the plates were turned upside down. And then the plates were incubated at 37 degrees Celsius for 24 hours before assessment. And as a result, the observed closure surrounding the disc was measured in terms of its diameter. This is the result for phytochemical analysis. This table shows the phytochemical analysis of dilute-indum fruit shell from dilute-indum ethanolic root extract, dilute-indum ethyl acetate fractions, and dilute-indum butanol fraction for phenol, flavonoid, saponin, tannins, and alkaloid. From this table, the triple plus symbol represents the large amount of phytochemical component, while double symbol of plus represents the moderates of components. Single symbol of plus represents a small amount, while the negative symbol represents the absence of phytochemical components. Based on the phytochemical analysis result for the phenol, the existence of phenolic compound in DIECE, DIEAF, and DIBUF can be associated with their polarity and solubility. For flavonoid, they were found in DIECE, DIEAF, and DIBUF. For saponin, DIECE, DIEAF, and DIBUF shows the absence of these components. While for tannins, different levels of tannin in extract and fraction show that there are a variety of polarity that presence of tannin which reflect their different solubility in the solvent system. And lastly, for alkaloids, they were also found in every extract and fractions, implying a broad range of chemical composition and solubility. For the results of antibacterial activities, this table shows the average measurements of the zone of inhibitions of the indum fruit shell extract and fractions against S. aureus and E. coli. According to Fatiha et al. On 2022, the zone of inhibition were classified as weak if the diameter less than 5 mm, while for the diameter from 5 to 10 mm classified as moderate, 10 to 20 mm classified as strong, and lastly, large than 20 mm classified as very strong. Based on this result, for S. aureus bacteria, the dilute-indum ethanolic crude extract show the strong zone of inhibitions, while for E. coli, the strong of zone inhibition shown by the butanol fractions. Based on the antibacterial activities result, polar protic solvent, which are ethanol and butanol, extract more effective antibacterial compound from the polar aprotic solvent ethyl acetate. DIECE exhibit the largest zone of 12.67 mm against S. aureus, while DIBUF exhibit the largest zone of 11.33 mm against E. coli. Phenols and flavonoids are more soluble in polar solvents, explaining the higher activity in DIECE and DIBUF. As conclusion, study on antibacterial activity and phytochemical analysis of dilute-indum fruit shell extract reveal bioactive components such as phenols, flavonoids, tannins, and alkaloids. The results show significant inhibitory effects against Staphylococcus aureus and Escherichia coli. The D-indum ethanolic crude extract, ethyl acetate fractions, and butanol fractions all exhibit different levels of antibacterial activity. As recommendation, further research on the specific mechanism underlying the antibacterial activity of the indum fruit shell extract is recommended due to the comprehensive finding of this study. Other than that, clinical research might also be pursued to assess the extract's safety and effectiveness in a more intricate biological environment, which could result in the discovery of new antibacterial agents or complementary treatments.